Hi, everyone. After our most recent Ask Me Anything episode of Translating Proteomics, we realized that there were a lot of people excited about the Nautilus platform who had lots of great questions about how it works, its potential, and more. So, thanks for joining me today on our very first episode of Parag Answers Questions about the Nautilus Proteome Analysis Platform. Our first question has to do with the probes that we use in the platform. In particular, were there were questions about how do we design and choose our probes? How do we know how many probes are necessary for proteome or proteoform coverage? Uh, Another question related about how easy it is to make new probes for new organisms, new proteoform families, and particular types of PTMs. So before I dive in to answer the specific question, I want to take a step back to just give a very quick overview of our underlying method for proteome and proteoform analysis known as iterative mapping. In this method, it has really three steps. In the first step, we attach millions to billions of individual protein molecules from the sample to distinct landing pads on a massive, hyperdense single molecule array. Each protein gets its own landing pad and can be spatially, optically distinguished. Really, in your head, you should think about gluing each molecule from the sample down to a coordinate on a giant chessboard. Once we have those proteins glued down, are able to probe these millions to billions of individual protein molecules over and over and over again with a series of affinity reagents that have been fluorescently labeled. We use different probes in each cycle of analysis so that every time you touch a protein, you're getting a little bit more information about which protein is present at a particular coordinate on the array. And so when we see landing pads light up, we know that a probe bound. We then take all of that information, put it through a pretty sophisticated machine learning algorithm to analyze the composition of that particular molecule. And based upon what we see and say, oh, we observed a pattern of binding of probes 1 and 3 and 6 and 87, and we know what those probes bind to, we can then infer the identity of the protein at that coordinate or learn about what the proteoform is that's present at that coordinate. So, what's different in studies of the proteome versus targeted proteoform studies? In studies of the proteoforms, we use extremely specific reagents that recognize either a particular protein or a particular modification site within a protein, such as a phosphorylation on site 181 of tau. In proteome studies, we instead use a very different class of affinity reagents that we call multi-affinity probes. These probes are probes that we have designed and built in-house that recognize short amino acid sequences, typically 3 to 4 amino acids long. They don't tell you what protein you're looking at on their own. Each one is not specific to any particular protein, but they give you a little bit of information about an epitope that is present within a protein. And cycle after cycle, we build up increasingly more information about which epitopes are present in a protein at a coordinate on the array. And that machine learning algorithm then asks the question, given this pattern of observed epitopes, what protein must this be? To determine how many of these multi-affinity probes we need to identify or distinguish every protein in the proteome, we assume certain levels of on-target and off-target binding, and we know from the protein databases the sequences of all the 20,000 proteins encoded in the human genome, or 70,000 splice variants are coming from Swissprot, and we calculate how many unique probes with these binding characteristics are necessary to discriminate amongst nearly all the proteins in the proteome with high confidence. Our models suggest that we need around 200 to 300 multi-affinity probes to map substantively all of the proteome. For more detail on this modeling, we've described it in extreme detail in our preprint by Egertson et al. On bioRxiv, and talk about some of the assumptions that go into that modeling in terms of the on-target binding, off-target binding, non-specific binding, etc., as well as how many proteins a given probe recognizes, how many epitopes a given probe recognizes, why we opted for probes that recognize epitopes of length 3 versus 5 or 6 or 7. Now, that's one piece of the question of how do we design and choose our probes. In general, what for proteome coverage, we're looking for probes that recognize a large number of proteins. Ideally, in any given probing cycle, a probe will recognize somewhere between 10 to 20% of the proteins in the proteome. That's driven not just by what epitopes something can recognize, but also by that probe's fundamental kinetics. And so that gets to how do we characterize our probes? And this is something where we do a just tremendous amount of work. We start by first using phage display or similar techniques. We've also used animal models and others to select antibodies or aptamers that recognize particular epitopes. We then verify that after we have generated a probe candidate that it does in fact recognize the epitope that it was raised against. Next, we expose that probe using a technique called FIPSeq and similar methods to libraries of millions of different peptides that cover the human proteome to learn more about a specific pattern of epitopes that a probe might bind. This accounts for the fact that you might raise a probe to, for instance, the motif tryptophan, asparagine, lysine, and it might bind to epitopes that are not just tryptophan, asparagine, lysine, but are, for instance, tyrosine, asparagine, lysine, with some level of binding strength. And so really that step of exploring how a probe binds to the diversity of epitopes that it might encounter is a critical step. And we build probabilistic profiles that describe the likelihood that any given probe will bind to a diversity of different epitopes. The next step we take is we also do a number of kinetics steps. We use techniques like biolayer interferometry or SPR, surface plasmon resonance, to characterize what is the binding strength, what are the KDs, what are the on rates and off rates of each probe. We also test it on our platform itself to understand how well does it bind to our protein that's been presented on our nanoparticle. We also characterize how much does it bind to off-target areas of the flow cell. Does it bind to the areas where there are no proteins? Hopefully not, and we validate that that's true. One other step that we take is we verify that we can remove it. So using our standard remove buffers, which, for example, for a long time, we've used things like 10% SDS to strip off the antibodies cycle after cycle. We want to make sure that the antibody is able to bind, but also that we're able to get it off. So a tremendous amount of work goes into characterizing any given probe. In terms of making new probes for new organisms, I'll just point out that our probes recognize short epitopes, 3 to 4 amino acids long. They are not protein specific. So really, there isn't anything that's needed to be done to make new probes for new organisms, other than characterizing what is the likelihood that a given probe will bind to a different protein in that organism. And so long as we have a reference database, we have underlying models that help us understand what's the likelihood of a probe binding to a particular protein, a given epitope. And so the Nautilus platform should ultimately be quite general and apply to a diversity of organisms. I'll just come back to proteoforms and for targeted proteoforms, our process of characterizing probes is similar, but just a little bit different than for multi-affinity probes. For targeted proteoforms, again, we're looking for very specific probes. So we are very interested in characterizing on-target and off-target, just like with multi-affinity probes. We're interested in characterizing kinetics. But in general, we do a lot of work with synthetic peptides, epitopemers, where we concatenate multiple peptides together. We also make synthetic proteins and modify them so that they present particular sites of modification. And so that work allows us to validate that a given proteoform probe is actually recognizing, again, that specific site at 181 and is not cross-reacting with a phosphorylation at 214 or 217. We additionally do work to see how adjacent modifications might impact binding. For instance, does the phospho 214 recognizing antibody on tau bind equally well when there is or is not a phosphorylation at a site 217 nearby? So a lot of this work is done with longer synthetic peptides or specially made proteins that have known phosphorylations. So while it is not necessarily hard to import new antibodies, we do a lot of work to make sure that our antibodies are extremely specific, they bind well, and that we understand their binding characteristics. If you'd like to learn a little bit more about our proteoform efforts and our characterizations of that work, I suggest you check out our in-depth look at our methods in our June 2025 Jolie et al. Preprint that demonstrates our single molecule analysis of tau proteoforms across model systems and patient samples. Just to sum up in general, we have put a tremendous amount of work into building a process to deeply, deeply characterize our probes to make sure that when we're measuring them on platform, we understand what they are measuring and when they're indicating differences between samples, we really have a strong molecular foundation for trusting those differences. Uh, in general, getting excellent probes for iterative mapping of proteomes and proteoforms is a high priority for everyone inside Nautilus. Our next question asks about how does the limit of detection and the limit of quantification of the Nautilus platform compare to other technologies? First off, I want to say that this is an incredibly deep question, and it's important before diving in and to really take a step back and define what do we mean by limit of detection and limit of quantification. For a technology that identifies or quantifies anything, the limit of detection is the lowest amount of an analyte that a technology can confidently report the presence of an analyte. However, even if a technology can qualitatively say, oh, this analyte is present, that does not mean it can accurately or precisely report how much of that analyte is present. For instance, there might be a wide range of concentrations that for some analyte, which the technology gives very similar readouts. It can tell you that it's there, but it can't accurately tell you how much. And you'll often see limit of detection, limit of quantification described in titration series curves where you vary the amount of an analyte under interrogation and you look at the signal response on a platform to how much of that, how much is being read out, what's the report, whether that's an intensity, an area under the curve. There's some measure of abundance. And typically those curves are S-shaped. And when we talk about the limit of detection, that might be towards the bottom of the S. Whereas the limit of quantitation, we often are really referring to the linear range, whereby if I change the amount of an analyte by X amount, I get a linear response in my estimate of the abundance. Now, one other related factor that people often talk about is dynamic range, often in the same breath as limits of detection. And so, depending on how people talk about dynamic range, that can be across analytes. So I can measure an analyte that is present at this abundance, and therefore my dynamic range is the range between those two. We've spent a lot of time characterizing and calculating how limit of detection is impacted by the number of intact protein molecules measured. across arrays of 100,000 molecules, a million molecules, a billion molecules, et cetera. For a deep dive into that, I suggest you take a look at our Eggertson et al. 2021 preprint. And that work suggests that for our arrays that measure on the order of a billion to 10 billion protein molecules per experiment, we should be able to quantify, not just detect, but quantify proteins over about 9 orders of magnitude range and abundance. As a single molecule platform, our theoretical limit of detection is theoretically 1 molecule. And at the single molecule level, the limit of detection is theoretically the same as the limit of quantification. And our early efforts at pushing on that have demonstrated sensitivity down into the octomole range, which is on the order of tens of molecules. What what creates a floor for a single molecule assay is its false positive rate. As we as your false positive rate goes up, then you can no longer confidently quantify whether there are 1 or 10 or 100 molecules, because those might be false positive identifications. We've done a tremendous amount of work in limiting our false positive rates. And because we have so many measurements of each protein molecule, we have the ability to turn a knob on how confident we require our identifications to be. And that further allows us to push down to having a false positive rate that is vanishingly small. Um, that's work that's actively ongoing, and it's going to get better and better the more cycles we run, the more probes we use to identify proteins. But in general, how that compares to other platforms, well, when one thinks about in mass spectrometry, for instance, the amount of protein that's loaded on column is somewhat analogous to, in our single molecule platform, how many molecules are loaded on our flow cell. Typically, what you're estimating is that the limit of detection is in maybe that femtomal range typically, and with a tremendous amount of work in the high attomole range. But on our platform, we've demonstrated the yoctomole range, so several orders of magnitude better than can be observed using traditional platforms. As compared to ELISA, or again, typically we would expect that our platform would be several orders of magnitude more sensitive than a traditional ELISA. Keeping on that theme about comparing our method to existing methods, I also want to comment on the other application of the platform, which is to mapping of proteoforms, not just proteins. In our recent preprint, we demonstrated a quantitative dynamic range of more than 3 orders of magnitude and a limit of quantification of roughly 0.1% relative proteoform abundance. And what that means is that we're able to quantify accurately proteoforms that make up as little as 0.1% of the total tau abundance within a sample. And to be clear, this is just the beginning of where we anticipate going. But I encourage you to take a look at that. We In doing that study, we were able to measure over 130 proteoforms across a range of different biological samples, cell lines, organoids, as well as mouse brains, human disease tissue samples. And so the comparator, what can be measured with other samples, is a little hard to say. They're really ELISA, other affinity-based methods simply aren't able to measure proteoforms. And even studies using top-down mass spectrometry, quantifying limit of detections out of complex biological samples is a little hard to assess for quantitative accuracy. So we believe that this is a really unique and special measurement that should open up a lot of very exciting biology questions. We don't have a lot of time left today, but I'll answer one last question, which is really just about the availability of the platform. Many of you asked when it will be available, how will initial partnerships or collaborations work, how can people access it, how much will it cost? And I think right now we haven't announced any information about that. However, what I can say is that we are actively seeking deep collaborations and deep partnerships with people. Really, this measurement, particularly of proteoforms, is so new that what is really required are some deep foundational biologic studies that help illustrate the value and impact that proteoform analysis can have on understanding mechanisms of disease, understanding disease progression, trying to identify novel biomarkers. And so we're constantly reaching out to people and listening about potential partnerships. And really, what we're looking for are really deeply knowledgeable investigators who have exciting questions that they want to use proteoform analysis to help answer. And so we very much encourage you to reach out and send us a note and say, hey, would you like to partner on this? And we'd love to talk to you.